Today we are going to start talking about frog development and we are going to talk about fate maps. Why we're going to talk about frog development? Because this type of frog, the leopard frogs, produce giant eggs, and those eggs are so big that they were very useful to understand morphogenesis in the beginning when there was less genetic tools and less available tools. So here is one of the first articles to show how each one of these cells were divided. Okay, let's start with this, and I hope that get ready to review some of the terminology that we talked about: blastula, blastocell, archanteron, all these blastopore, all this terminology that we talked in previous classes, and hopefully now you can use it to understand much more what is the frog development and what is fate maps. We're going to start talking about what is a fate map, then we will talk about all these stages of the frog life cycle in a very general way. And during the semester, we are going to talk about each one of these in much more detail. Then we will talk about some applications of the why we study frog development and what it was studied in the past. And we are going to talk about some techniques that they were used to study fate map. And in developmental biology, I remind you that the idea is that the cells have to be very big when they were not stains or genetic tools. So maybe you will hear about different organisms that you never hear. So it's very common that we talk about sea urchins. So in your textbook, you will find information about them. Ascidians, very important model organisms for developmental development, and the one that we talked in the previous class, C. elegans, that is relatively new. If you remember, one of the advantages of the C. elegans is that it is a transparent nematode that is about 1 millimeter. That, although for us it looks very small, for microscopy is amazing, and it produces a small number of cells. And here you can see, as you watch in the previous video, each one of these are color coded, all the regions. So here you have in green the intestine, the nerve rings, the germ cells, the muscle. Each one of those structures we can produce. We know when they appear, in what, from which specific cells, and we can track each one of the cells, the formation of these structures. So this is what we were talking. So when we talk about fate of each cell, so we can map them out in the embryo, and process is called cell single lineage. So with the fate map, with the C. elegans is perfect because there are not too many cells. I know this look like a lot, but with other organisms this will be impossible. So here you can see all the muscles. Here are type of muscle, the endoderm, the neurons, all these structures we know in where they appear, where they come from, and we can know from a single cell when they appear. So this is pretty useful. And now I'm going to show you, we have this, and they do this also something similar for cancer cells, but only from a genomic perspective. It would be very difficult to do it, but from a cellular perspective, but we can also produce this kind of cell lineages at the genomic level. Okay, so now we're going to talk about how they start doing this type of studies using the frogs. Before that, I forgot to tell you that I was going to talk about the sea urchin. So here is a diagram from your textbook. So here is 16, the cleavage, gastrula, and here is the larvae. And you can see that there are 3 germ layers, in 3 different colors. And you can see how they start differentiating which one is going to form the ectoderm in blue, which one is going to form the endoderm that is going to have the midgad and the hindgad, and here which one is going to produce the endoderm. So that is going to be around here. So C. elegans, sea urchin, ascidians are great organisms to do that. So what is a fate map? So here you can see the beautiful ascidians, and we can do the same thing with the embryo of the ascidians and say, like, This is the initial stage, and look, all of these cells, the green cells, are going to be located here. They start here, and you can track that. So what is a fate map? A fate map is a method used in developmental biology, and why we do it? To study the embryonic origin of various adult tissues and structures, and the fate of each cell or group of cells is mapped onto the embryo, showing which part of the embryo will develop into which tissues. When carried out at a single cell resolution, this process is called cell lineage tracing, as I show you with the C. elegans. And I also told you that it's also used to trace the development of tumors. But I told you that in the case of the tumors, unfortunately, we cannot do it at the cellular level. We can only do it at the genetic level when the tumors already have invaded, yeah, or move into the other tissues and try to see when they started and what are the relationship of those. Okay, now that you know what is a fate map, we're going to talk about the fate maps done in the frog. That was one of the most important systems and one of the first one that we use. So we're going to talk about the frog life cycle, and we're going to start with the first stage, that this is the fertilization. Okay, so the frog life cycle has 6 different stages. This frog is called, this is the scientific name, so it's also called the leopard frog. And as I said before, they have giant eggs, so they are used for research a lot, especially in the past. And these are the 6 stages that we are going to discuss. And here are all the stages taken, the diagram found in your textbook. The first stage is the stage of fertilization. So here you can see when the sperm and the oocyte fuse together, and so these mature cells, they get together and fuse to form a zygote. So here is the process of reproduction. Here, and remember that we have been talking about preformation and epigenesis, and here I want to tell you that not only the genes are important here, so the process of fertilization of the leopard frog that I said to you that it was one of the model organisms, the environment play a key role. Why? Because if they don't reproduce at a specific time, they can run out of food. So for that reason, it is important that external environments like the temperature, like the amount of light, yeah, all these signals will produce signals in the body that affect hormones like the pituitary that secrete hormones, so the gametes mature and they reproduce at exactly the right time. Okay, so that is important. So we think that genes are important to create all these process of reproduction, but also the external environment also has an influence. So remember, the life cycle is influenced by external factors, and they need to be seasonal events, so they reproduce at the exact time, so they don't run out of food. And in the lab, those factors they need to be taken into account, so they produce eggs. So here is where I told you about the different hormones that they need to be secreted. And here, the pituitary, that is influenced by the photo period, the amount of light, the temperature, and that it will affect the pituitary gland, and it will secrete hormones that in turns will allow the gametes to grow and mature. In the lab is through injections, so you inject the frogs with a specific hormone that allow the hormones, the eggs to be released at a specific time. Okay, so we talked about fertilization. Now let's talk about cleavage. So in cleavage, you can find it here. So the fertilization is done, and what happened here in this stage, if you see, is that the zygote start dividing, and it goes through a morula. That, if you remember, we talked about the morula stage in the previous, and the blastula. Yeah. So remember, morula is this stage Morula came from a word in Latin that it means like berry. In Spanish is mora, mora. That yeah, so that have like all this. It looked like a soccer ball, yeah, or like a berry. And the blastula looked is a hollow ball, yeah. That so inside it became empty, yeah. So it began with the first division and it ended up with the blastula. Okay, so what is the cleavage? The cleavage is a series of rapid mitotic divisions that happen after fertilization, and the large cytoplasm is divided into smaller cells that are called the blastomeres. Okay, so I'm going to show you now some these pictures of the morula and the blastula in more detail, so you remember them and you keep the concepts with you. So in the topic of fertilization, I want to tell you that there is an animal hemisphere and a vegetal hemisphere, and what it means. So here you see that this doesn't have anything to do with animals or with plants. What happened is that here in the vegetal pole is where all the food, all the nutrients are located. While here you see that the nucleus, the pronucleus will be here. So here is where there will be active divisions, and here is where the food will come from. Yeah, so you will find active division here, and here you will find subdivision, but less divisions here in the case of the frog. Okay, and so after the fertilization, what happened is that there is a stream of movements that to set up the egg to distribute the materials to create this animal hemisphere and vegetal hemisphere. Yeah, and there are three main sections: the animal pole, vegetal pole, or animal hemisphere, that is and vegetal hemisphere. The animal hemisphere is the one that is pigmented, while the lower is the white vegetal pole, and in between those there is one that is not pigmented, that is the gray crescent or the marginal zone. Okay, so remember, you will see the difference in the following slides about how in the frog you will start finding that the cell division, that the presence of the nutrients here and the rapid cell division will create a distinct size of the cell division. Yeah, so let me show you in the following slides. And now here you can see the morula again, and here you can see much bigger, and each one of the cells of the morula are here, the blastomeres. So remember the morula, that in Latin is morus, that means mulberry, yeah, is an early stage of the embryo consisted of a solid ball. So at this point, all the cells are inside, yeah, and each one of these cells are called blastomeres, yeah. Now, when the next step, we're going to fill this ball with fluid, and all the cells are going to be outside, and the ball is going to be empty. So look, zygote, we just talked about the morula, yeah, 8-cell division, and now look, the blastula, and when you cut it, the cross section, it will show that here is empty with a fluid, and this cavity is called blastocell, and the cell now is called blastoderm. Okay, so just as I remember, blastula is a hollow structure that is formed after the zygote go under cell divisions, and the blastula consists of two main consists of blastoderm and blastocell. The blastoderm is the epithelial layers, so the cells, and the blastocell is the fluid-filled cavity enclosed in the blastoderm. Okay, so I know that I'm giving you a lot of terminology. And so in the previous class, we talk about this. Now we talk about this, and hopefully, slowly you will start remembering. Sometimes they ask these questions in MCAT questions, sometimes not, but at least hopefully this can help you to understand this better. Okay, and here you can see the pictures of the article, and here you see we have been talking about the animal hemisphere or animal pole and vegetal pole, and here you see the first division, here the second division, and here the third division. And So we're going to talk about that, okay? And I want that you think, okay, are the division north and north to south? Are the division west to east? Where are the divisions? A vertical, transverse, oblique? Where is there a pattern there? And this is something that you was studying and always occur in the same way. So let's talk about that in the following slide. So after 2 or 3 hours after fertilization, the zygote began to divide and and repeated division in successive fashion is known as a cleavage segmentation. The division is mitotic. Yeah, and here first, and the first one is north to south. Yeah, the second one again is north to south, and now you have 4 cells. And when you have 8 cells, you cannot see the 8th one because it's in the back. This one is the one that it goes to from west to east. Yeah, that is not is a transverse cell division, and if you can see here, the cells are much smaller than this one that are the bigger ones, and you will see that this will continue like that. Remember, here will be most of the food, and here will be most of the cell division. So as I just told you, the first cleavage is vertical. You can see it here in red, and this is the 2-cell stage, and it starts with a small cleavage at the animal pole that gradually extend surrounding the zygote that divide into cells. So it start here in the animal pole, and it continued there. The second cleavage is similar to the other one, yeah. And here the cells, as we said, they are still called blastomeres. Now we're going to the first horizontal cleavage, so they are not anymore vertical. And as I said before, they are not in the equatorial zone. They are cells that don't have that they have an equal size. And this one, there's small ones are called micromere or epiblast, and the big ones are called megameres or hypoblast. Okay, and here you hear this one will have a lot of yolk, and this one will be yolkless. So we are still here in the morula stage. Sorry that in the beginning I told you that we go zygote, morula, blastula, but we are still in the morula stage. We are just seeing how this first cell division happened, and I show you now that the micromere and megamere appear. And here again, you can see in red all these micromere and in blue all the megameres, and the difference anatomical and also the nutrients that they have. That is what creates the difference. Something that is important to tell you here is that these divisions are called holoblast, that they include the entire embryo. Yeah, so although we show that they were on an equal size in the cells, they go all over, they go from the top to the bottom, from the top to the bottom. Yeah, and these are called holoblasts. There are other ones, like for example in fish, that the yolk is below, and they just use that yolk for as for nutrients. They don't use it for cell division. So those one are called meroblastic. Yeah, so it's an incomplete or partial cleavage, meaning that the egg doesn't penetrate. The cleavage doesn't penetrate completely the egg, as you can see here. And there are more examples. So I'm not going to ask you to memorize all those things, but if you can see, there are a lot of type of holoblastic cell division. You can see that they include the entire zygote. Here the one that the mesolithical. Sorry, I don't even can pronounce some of these ones. But one that grabbed my attention is the one that I study that a lot, that it was the one from Drosophila, or that I hear a lot, that in this case you see there is one embryo and there is one nucleus. Now there is one embryo, one single cell, and now it's 4 cells, and they continue doing this for several cell divisions, and there is still one cell. Yeah, so here they don't even have cell divisions. Yeah, they divide only the nucleus, and then the nucleus migrate to the end, to the periphery, and they form the cells. And so, in other words, I'm showing you one way, the one that the morula and the blastula is formed in the frog, but during development or during evolution, there are many different ways to do that. Yeah. And I'm not going to ask you to memorize every single of those. There will be practice questions that help you to memorize some of those and show you some of the things that I key to remember. Okay, so now here you have the stage number 7 when you have much more number of cells. Yeah, you have 32 cells, and look, later there will be much more cell divisions, and there will be a process of tissue involution that we will talk about that in a second. I will show you a video of the process of gastrulation, so it helps you to understand much better what is that. And again, involution is a term that we talked before in previous class. Hopefully you can use it here. Okay, so now we are at the stage of 32 cells. And here start with a vertical, and they go to 2 horizontal cleavages to give this number of 32 cells. Okay, so here it doesn't make sense anymore to count in which way the cells are divided. I just want to show you that, or what they start studying is that it always happens in the same way. And later there are so many more cells that other processes are taking place. Yeah, now it's not only cell division, it's now there's tissues that start moving, and so let's start talking about those. Okay, so we just talked about the fertilization, then we move to the cleavage that start with the zygote and end up with the blastula. Now we're going to move into the gastrulation. Now we're going to start studying gastrulation. So here in gastrulation, we already have a blastula, and a blastocell is going to start forming, and you will see a lot of movements, and we are going to end up with the 3 germ layers that we talked in the last class. Yeah, remember ectoderm, endoderm, and mesoderm. Okay, so what I want that you remember from gastrulation is that gastrulation is defined as an early developmental process in which the embryo transformed from a blastula to a gastrula. So I told you here, we start here with the blastula, and we're going to end up with the 3 germ layers. So the blastula will have one-dimensional layer of epithelial cells, while the 3 germ layers is a multi-layers that have multidimensional structures. At the end of the gastrulation, we still not have organs. Yeah, we just start forming the tissues. Okay, and I wonder, do you remember these 3 key things? Yeah, for the gastrulation. In gastrulation, so as you can see here, so gastrulation start here. You see the invagination of a tissue here that you will start looking different. Yeah, so that you will form the endoderm, and the one outside in blue is the ectoderm. So you will see, if you see, there is one that is blue and one that is yellow, because there is a differentiation in the cells. The cells start differentiating, and when you go from a monoblastic, that it was in this diagram, the cells were all brownish, to a deep holoblastic layer. Yeah, so you have the blue one and the yellow ones. And you, in the case of the frog, start the formation of the 3 germ layers. Here is a non-exhaustive, yeah, there are more movements, yeah, of tissues that you will find during the process of gastrulation, and during this course, I just wanted to summarize some of those. So first one, here you go, is the process of involution. As if you can see here, you have one layer that is going to bend and is going to go under, and this will be involution that you will see in the frog in few seconds, yeah, in the following slide. There is another one that you will also see that I already actually talked about, that is the invagination one. A group of cells, in this case in blue one, are going to go internally, all as a tissue, and this is happen, this you saw it for the formation of the three germ layers that the blastopore is going to form, and the cells are going to invaginate. You haven't seen this one yet. It's called ingression, that instead of a tissue or like in the invagination, you have single cells that are ingressing. That is a process of delamination, that they go away from the epithelium. The other one is the epiboly, that in the epiboly you see that this is sorry, this is going to take some time. This is yolk. Yeah, the yolk is there. And the cells are going to go over and cover the yolk. Okay. And finally, the convergent extension, that you will also see it there, that here you have a narrow and short tissue in the beginning, yeah, and you're going to make it into a long, yeah, into a very long tissue. So you're going from 3 layers in this case into a one that is much longer. Yeah. So sorry, you go from one that it was wide and short into one that is long and narrow. Okay, and you will find that one also in the gastrulation process of the frog. And this process is called convergent extension, and you say the intercalation of cells. Okay, again, there are more processes that you can see there, but this one, I want that you keep it in mind so you remember it, and you can understand some of this concept much easier. So now let's give a first look to the process of gastrulation. So here you have your blastocell. Remember that this is empty, that this is the cavity that is full of liquid, and here you have the blastula, that is this empty bowl, and here you will see this tissue from a different color that is going to go and do an invagination, and is going to go here, yeah. And here is when gastrulation start occurring, and you're going to start forming this archenteron, that is the origin of the digestive tissue, yeah, the digestive organs, and you see that it moves, and depending on how far it moves, you can be uh, uh, if you remember the tissue, you will have an anus and a mouth, or you will have only one hole. If you watched the previous video, we talk about that. And so here you already have the blastopore, yeah, and you have two tissues, the ectoderm and the endoderm. And then late gastrula, you will have the three tissues, yeah, the mesoderm, the ectoderm, and the endoderm. Okay, so all these things are happening during gastrulation, from early to late gastrulation. Now we're going to watch a video that will talk in more detail what is happening in the frog, but this is just an initial explanation of what is happening. Okay, just as a reminder of the previous video, we talked about the three germ layers, and here I'm showing you these three germ layers. So the outer layer, it will be the ectoderm, and from there the nervous system originates, also the skin and the hair cells. In the mesoderm, the one that is in the middle, the majority of the organs will originate from there, including muscle, blood cells, blood vessels, kidney, heart, and skeleton. And the endoderm, from that one, it originates the respiratory and the digestive tract, and as we talked in the previous slide. Okay, so just as a reminder, we will be talking about these three germ layers and are the layers where all these organs and structures form.
If we look at a cross-section of an embryo of the frog Xenopus, we can see that at this point it is a ball of cells with a fluid-filled cavity. The cavity is the blastocele, and the embryo is currently in the blastula stage of development. A blastula contains large yolk-filled cells at the vegetal pole and smaller cells at the animal pole. The three colors represent the three tissue layers that become defined early in embryogenesis. Yellow indicates endoderm, red indicates mesoderm, and blue indicates ectoderm. At the beginning of gastrulation, a few surface cells, called bottle cells, move into the interior of the embryo, followed by other surface cells. We can track the movement of cells into the embryo if we add dye to a few surface cells. The movement of cells into the embryo creates a lip, called the dorsal lip, over which sheets of cells continue to move inside. At the same time, the ectoderm extends around the embryo surface in a process called epiboly. As gastrulation proceeds, a cavity, called the arc enteron, forms while the blastocele progressively shrinks. The arc enteron is the primitive gut and is completely surrounded by endodermal tissue. The endoderm at the roof of the cavity originated from the outside of the embryo. The cavity is continuous with the outside via the blastopore, which eventually becomes the anus of the animal. As the ectoderm extends around the embryo, another set of bottle cells forms. These cells migrate into the embryo, and other surface cells follow them, creating the ventral lip of the blastopore. By the end of gastrulation, the ectoderm has surrounded the embryo. Endoderm lines the inside, and mesoderm lies between the two. Additionally, the fates of specific regions have become determined. The endoderm gives rise to the digestive and respiratory tracts and associated structures. The mesoderm gives rise to the skeleton, circulatory system, muscles, excretory system, and most of the reproductive system. The ectoderm gives rise to the skin, sense organs, and nervous system.
I think this video did a great job explaining what is happening. You can watch it multiple times. I'm going to put it in Populi, so you can watch it more times. But look, here is showing you in this section what is happening from the external perspectives or from a surface view, and you can see how from the animal pole there is this process of involution, yeah, that is covering all the entire region. I told you there will be more cell division in that area, and that in that covering everything. And in the cross section, you can see here in red how the mesoderm, start moving close to the ectoderm, yeah, and start forming the arc enteron. Okay, so now let's continue describing what is happening during the frog development. Okay, so we start describing what it was a fate map, then we move to the frog life cycle, and we cover fertilization, the first divisions, the process of forming the blastula, the three germ layers. But right now we only have the three germ layers, we don't have any organs. Now we going to start forming the organs, and this is the process of organogenesis. Okay, so now we are here in the process of organogenesis. We start with a tissue that have not a clear forms. There's no clear organs, and they start with a gastrula, yeah, and we're going to end up with a fetus, yeah, that it already have organs and they are functional. So there are many things happening here. We are not going to cover it in detail. I'm just going to tell you that that we start with these amorphous cells and we're going to end up with a complete organ. And I'm going to show you some of the processes that are happening to go from a gastrula into a fetus. And as I said before, organogenesis is a series of organized and integrated process. And here, if you see, there is not neural tube. Here we already have the beginning of a nervous system, yeah, that is already formed. And here you have the notochord. Here you have the digestive system form. And I'm not going to go into detail. I'm just going to give you a lot of general things that are happening here. And during the semester, we are going to cover then what is happening. So here, there are key processes happening. So the organs to form during the semester, I'm going to show you that there will be a lot of differential development and how this differential development will happen, because there will be different genetic signals and different mechanisms involving that will produce different movements. Yeah, remember all the movements that I told you? There will be movements affecting that, and they will create each one of the organs. Yeah, to create each one of the organs, and that at the end of of this process will be functional for the organism to for in case of the frog to start the process of metamorphosis. And here now you can see the process of gastrulation and how, for example, from the endoderm you start forming the foregut, the midgut, and the hindgut. I'm not going to explain the class in this class how this happened. I'm just going to tell you that it happened, and it involved different transcription factors and genetic signals to do that. And at the end of the semester, we're going to talk about some of those. And during the semester, we will talk about this structure called the notochord that is very important. Here you can see it here. That is a flexible rod that stimulates the process of neurulation, the production of the neural system, and it will cause a bending here. You remember that we talk about that the tissues move and break, and it will happen, and there will be the differentiation of the mesoderm, and you will form somites to produce bone, muscles, and other structures. Yeah, and all of them will come from these three germ layers. So here again, just general things I'm telling you that we will cover during the semester in detail. For me, it's important that you are able to identify where is the neural tube. Yeah, where are the somites, because later in the semester we will talk about the somites, that you know where the notochord, that the notochord, it will be stimulate, as I said, the process of the formation of the nervous system, where is the mesoderm, the acantharon, the endoderm, and the ectoderm, that the ectoderm is outside and the endoderm is inside. And this will help us later to understand how each one of these components will subdivide and start forming a particular organ. For example, one thing that it will happen is that the notochord will end up creating the neural plate, and is divided in three stages that are here: 1, 2, and 3. And I want that you give a quick look of what is happening here, and I'm going to show you in the following slides what is happening. Okay, so let's go first with the first. So what happened here is that the notochord is going to create a fusion, so that this tissue, that the neuroplate, is going to have an inward movement, and you can see it here. You see it here, and these two, the blue, dark blue, are going to, the ectoderm is going to start fusing. Okay, you can see it very clearly in the third stage. So you can see here, the tissue has completely formed, the neural tube has fused, and now is on top of the notochord. Now you have the somites formed, it is not anymore the mesoderm now, and here you have the spinal ganglion. Again, this is very general, and what I'm showing you is that all the movements that are happening that I describe you happen during this process. Okay, so now let's continue with the metamorphosis. That I'm not going to talk a lot about the metamorphosis, just to tell you some of the major changes that are happening. So the metamorphosis is the changes from the larva into the mature adult, and these are some of the key components. So here you see in A that is an aquatic tadpole, and here the frog is a terrestrial frog. They have tails, and you can see that the tail start disappearing until it's completely gone. The skulk is soft and yeah, it's from a cartilage, while in the terrestrial frog is a bony skulk. The diet changed, so uh, uh, in the larvae there are teeth, while in the muscle in the adults there is a tongue muscle. The intestine start changing from a long intestine to a short intestine, and the gills disappear, and they are large lungs. So these are some of the adaptations that they changed during development to produce um, an adult frog. And as I said before, this is not we are not during the semester we are not going to talk about this a lot, but this is a component that is important to understand. Yeah, not only the metamorphosis not only happen in frogs, it also happen in insects, and those one are in insects. I work in insects metamorphosis, and they are also key process to understand, for example, cancer. Okay, and finally, we finish the larval stages, and now we're the maturity, and now we start again with the gametogenesis, and the gametogenesis is the production of these germ cells. And if you remember, in the beginning we started uh, there is the production of external factors that are very important. So light is important, um, the and also the temperature is important, and that it will lead to a secretion of specific hormones. Okay, so now let's talk about some of the applications. What do you think that are the applications of studying this process? Okay, do you think, have you heard about anything about pregnancy tests? Have you ever heard how were the pregnancy tests in the past? Yeah, 50, 60 years ago, I don't know, 100 years ago. So as I said before, frogs are an important model for organism development. So we share a lot of tissues, a lot of genes, we share a lot of structures in our body. So, and one example of that is the pregnancy test. Yeah, in the past, uh, frogs were used for pregnancy tests. So what they used to do is they used to inject with in the urine of women that it will have if they were pregnant, they will have a correct hormone that it will create that the frogs in few days will be developing a lot of eggs. Yeah, why? Because we have similar hormones. This will not cause the death of the frogs. It will just create that it produce a lot of eggs. Yeah, and this was in the 50s, 60s. That was the way that pregnancy tests were used. Then this was used for female frogs. Then it was found that also males can be used to do something. Can be also used to do that. Now we use an immunochemistry technique, and now frogs are not used anymore for that. But it's just something interesting that I thought that it was an application that maybe you have here or maybe you never hear before. A very cool application is that we talk about cloning, and we think that clone happened really recently, but actually with frogs we talk about cloning since a long time ago. Why? Because as the eggs were very big, it was very easy to manipulate them. So one experiment, you can have a frog A, yeah, and you can remove the nucleus. So here is the cytoplasm and the nucleus, and you can remove the egg. And you can take another one, a mature one here with the skin, and just keep the nucleus, and you can fuse them together and produce a clone of the frog B. Why of the frog B? Because this one was the one that had the nucleus. So it was a cool way to show that there are factors that depend on of the nucleus, and there are factors that depend on the cytoplasm. So it was a great tool for research. And this is just one example of how they were used. So now let's talk about, I remind you what were fate maps and some of the techniques that we use to produce those fate maps. If you remember, all the thing that we study is because we were talking about fate maps, and I show you that we have done that in a lot of detail with the frogs. Now, and that during the semester, we are going to talk about how these structures forms. Now, I'm going to show you some of the techniques that they have been used. So in zebrafish, frog, mouse, and chicks, there are many techniques that they have been used. Yeah, so now we can, in the past, we used to, so as I was telling you about the fate maps, so we have different fate maps. Some in the past, there were some that include dice that didn't, the idea that the dice didn't kill the organism or they don't produce a side effect. So the more successful are the ones that didn't affect the organism. Now we're moving away from those ones, and we are moving into genetic ones. Yeah, so here you can see we can just put fluorescent markers with proteins in the genome that try to find that, again, produce low toxicity in the organism to see exactly what is happening and to be able to trace them. Here you see only green, but you can have multiple colors and trace different type of cells, and there is a lot of techniques. Yeah, so I hope that this is clear, and if you have any question, let me know. And see you in the next video. Have a good day and bye.